Biochemistry metabolism of purine and pyrimidine nucleotides. Learning outcomes. On completion of this topic, you should be able to 1. List the purine and pyrimidine bases. 2. Briefly describe the synthesis of purine and pyrimidine bases. 3. Briefly describe the catabolism of purine and pyrimidine bases. Basic structure of nucleotides. Asterisk, each nucleotide is made up of 3 components. 1. Nitrogenous base, a purine or pyrimidine. 2. Pentose sugar, either ribose or deoxyribose. 3. Phosphate groups, mono, di or tri. Functions of nucleotides. A segment of a polynucleotide chain of DNA. General structure of purine and pyrimidine bases. General structure of nucleosides and nucleotides. Introduction to metabolism of purine and pyrimidine nucleotides. Asterisk, the major site of purine biosynthesis is the liver, although the purine nucleotides are synthesized by most of the tissues. Asterisk, the major pathway is denoted as de novo biosynthesis because the purine ring is synthesized from different small components. De novo biosynthesis of purine. Asterisk, de novo biosynthesis of purine nucleotides begins with phosphoribosyl pyrophosphate, PRPP. Asterisk, purine ring is assembled on a ribose 5-phosphate molecule of PRPP. Asterisk, there are 10 steps in the biosynthesis. Overview of purine biosynthesis. Asterisk, the committed step in de novo biosynthesis is the reaction catalyzed by amidinotransferase. It is inhibited by AMP and GMP via feedback inhibition. Asterisk, PRPP activates amidinotransferase. Regulation of purine biosynthesis. Asterisk, this pathway ensures the recycling of purines formed by degradation of nucleotides. Asterisk, salvage pathways produce nucleotides from 1. Phosphorylation of free bases by phosphoribosyl transferase. 2. Phosphorylation of purine nucleosides by kinases. Asterisk, this pathway is of special importance in tissues like RBCs and brain, where the de novo pathway is not operating. Asterisk, in comparison with de novo pathway, salvage reaction requires lesser energy. Asterisk, purines are mainly degraded into uric acid in the liver. Asterisk, the most common abnormalities is an elevation of uric acid level in the blood, referred as hyperuricemia. Asterisk, uric acid level is increased in gout. Degradation of purine. Biosynthesis of pyrimidine. Asterisk, the pyrimidine ring is synthesized as free pyrimidine from carbamoyl phosphate and aspartate before the ribose is supplied by PRPP. Asterisk, carbamoyl phosphate is derived from carbon dioxide and glutamine. Regulation of biosynthesis of pyrimidine. Asterisk, the regulation step in pyrimidine biosynthesis in human is carbamoyl phosphate synthase IICPS Roman 2, the rate-limiting enzyme. Asterisk, this enzyme is inhibited by UTP and activated by PRPP. Salvage reactions for pyrimidine. Genetics, immunology, and molecular medicine lesson, metabolism of purine and pyrimidine nucleotides. Final products of pyrimidine degradation. Beta-alanine. Beta-aminoisobutyrate, negative NH3 and CO2. Unlike urate, the product of pyrimidine. Catabolites are highly soluble. Reduction of ribonucleoside diphosphates forms deoxyribonucleoside diphosphate. Ribonucleotides catalyzed by the ribonucleotide reductase complex. Biochemistry metabolism of purine and pyrimidine nucleotides conversion of DUMP to DTMP. Biochemistry metabolism of purine and pyrimidine nucleotides conversion of DUMP to DTMP. Biochemistry metabolism of purine and pyrimidine nucleotides. Biochemistry metabolism of purine and pyrimidine nucleotides. Summary. Asterisk. There are two types of nitrogenous bases in a nucleotide, that is purine or pyrimidine base. Asterisk purine and pyrimidine nucleotides can be both produced from scratch, de novo, or asterisk a 1 carbon transfer is required to convert uracil to thymine, catalyzed by thymethylate synthase. Biochemistry metabolism of purine and pyrimidine nucleotides.